Hi everyone, I'm Ashleigh Davies. For those of you who don't know me, I'm a second year PhD student working with Sarah Morgan in the departments of biomedical computing and early life imaging. So I did my undergrad at Oxford just in general engineering, and now I'm working on like neonatal brain development from a sort of computational perspective. So today I'm going to be talking about data visualization and connectome visualization. So it's kind of a slightly side project from the work that I'm sort of doing day to day, but I think it's a really interesting thing for us to talk about, especially kind of in the first month of the academic year. Nice to have a refresher on some of these things. Okay, so let me just get on. Okay, brilliant. So I'm going to start by, if I just hide this. Bear with me a sec. Here we go. Okay, so I'm going to start by going over kind of the history of data visualization and why it's been important kind of up to now in terms of scientific research. Then I'm going to run through some of the key plotting libraries. This is probably going to be a recap for some of you and maybe an introduction for some of you. I'm going to talk about how to do things like color customization when you plot data. I'm going to go over some of this person who I'll introduce, Edward Tufte, and his design principles, which I think are really important. Then I'm going to move over to talk about plotting kind of on relevant anatomy. So maybe when we want to plot data on the heart or on the brain, depending what your application is. I'm going to touch on connectivity circles. This is one way to plot connectomes. I'm going to go over an example using PyVista to plot the brain. And then I'm going to move on to talk about communicating research. So things like poster presentations at conferences and also designing pipeline flowcharts. So when we do a study design, how to display that using a pipeline. Okay, so this will be a recap of, how do I move from one side to the next? So this is just a recap of the abstract that you might have seen that was emailed round. So basically, why data visualization is important. It covers kind of the full pipeline through research. So maybe if you're doing a literature review, you can use data visualization to understand what the different areas of literature might be relevant. Then when you come on to design a kind of methods, you can use data visualization to design pipelines, analyzing results, and ultimately conveying your research to audiences, which is really important. So the kind of learning objectives of this presentation today is going to be to signpost some available resources, so things like plotting libraries, poster design ideas, also to provide generally kind of creative ideas for displaying more complex data, so not just two-dimensional data, and finally to really impress on you the importance of the visual side to research. So the history of data visualization is really quite interesting if you look into it. So to start with, the kind of early data representation is just in the form of basic line plots. So traditionally for things like economic data, so maybe sales, etc., it's just basic hand-drawn line graphs, bar charts, and pie charts, and so on. Then by the time we get to roughly 1858, which is during the Crimean War, Florence Nightingale kind of produced this really interesting plot, which is the coxcomb diagram. So what this represents was actually mortality rates through the year of 1854. And in green is basically the proportion of deaths that came from the war directly. So this is a really nice example for me because I think it's one of the early examples of data representation being used for advocacy. So the idea of this plot was to represent that, this is a really high proportion of the population dying as a result of war compared to other means. And she just really wanted to stress that, this has to stop. It's not sustainable. Ultimately, for the population, it's going to cause problems. And I think it's nice for us to reflect on data representation as a kind of way that we advocate for the people that we conduct research regarding. For example, to stress to audiences the prevalence of a disease, we can use data to represent that, or just to advocate for the kind of necessity of the research that we do. So I really like this example of that. by the time we get to the kind of early 1900s, we start to have more automation of data representation. So things like oscilloscopes, they will record using pen, but it's the kind of automated arm doing it. Then we obviously get the introduction of computers, so that made a massive difference to the way that we can kind of represent data and plot things. But early computers were very much monochrome. So what are some ways that you can represent data when you don't have the benefit of color, for example? Any ideas? Yeah, exactly. So things like shape, also like black and white, contrast. Then by 1980s, we have like full color capacity, so then we can represent data in many more kind of dimensional ways. Obviously, now we have loads of tools like MATLAB, Excel, everyone's favorite Matplotlib. And this part is kind of interesting because I was thinking of a somewhat amusing example of what data representation could be like in a kind of futuristic scenario. But in reality, what I've come up with is not really that far off from what we can already do. So, for example, immersive datascapes and data representation in virtual reality. This is actually something that is being launched in various fields. For example, in the kind of context of pain research, sometimes virtual reality is being used to kind of represent pain in a more three-dimensional way and to understand it like that. So this is kind of where we have got up to today. So some of the plotting libraries that we have available now, one example is this matplotlib. So any of you that have backgrounds in computer science or use plotting in your undergrads, etc., you will understand that these are kind of the basic plots and data representation styles that we have available. I think it's nice to recap the basics for the purpose of understanding when we want to use what kind of plot. It's easy to get your data and just plot everything in a scatter diagram and then have a look. But in reality, what we want to actually show about our data reflects the plots that we should be using. So just to really quickly recap some of these kind of basics, when we use a line plot, it's for a situation where we already know the relationship between our variables. So to plot ground truths, for example, or to plot a kind of physical construct, it's appropriate to use a line that connects the data for all values of x and y, for example. When we have experimental data, we need to use a scatter plot so that we can kind of draw conclusions from maybe what the best fit line is going to be and things like that. A bar plot is obviously for representing frequency, but this will be when we have discrete data. So, for example, categorical data about population groups and so on and so forth. When we want to use a histogram is when our frequency is continuous. So something like height, yes, we can bin it into bars like this, but we also need to use a kind of continuous histogram type outline. Then we have a box plot. This is a really nice way to represent a kind of summary statistic of your data. So if really the part of your data that matters is the distribution, for example, the median, the interquartile range, a box plot is a nice way to avoid confusing everyone with the full histogram. So it's a nice way to compare multiple histograms, and it's just really a summary data. When we would use something like a pie chart is when we want to represent kind of proportions. And this is a bit similar to what the coxcomb diagram does as well. Okay, so then we have a few more kind of rare options, such as an error bar plot. This is a really nice thing to use if what you're trying to impress on your audience is like the measurement uncertainty, or if measurement uncertainty is really relevant in what you're displaying. The fill between you don't really see very often, but it's often used in a kind of economics type applications where you want to represent whether you have a kind of net positive or a net negative. We can use like the area tool. And also we can plot images on Matplotlib as well. Okay, so another similar option for some of these applications is Seaborn. Some of you might have used Seaborn if you plot heat maps and things like that. I would say it's similar to Matplotlib, but it has a slightly more kind of finesse statistical standpoint. So for example, not only can we do line plots, but you can also plot a kind of error band, if you like. So some of you might end up using something like this if you're plotting normative models or trajectories that have a kind of error field around them for different kind of parts of the population. Similarly, adding error bars onto a kind of bar chart is really useful to look at measurement uncertainty and things like that. One plot that I really like that you can do using Seaborn is this violin plot. So it kind of builds on the concept of a box plot, but you can actually plot the distribution alongside. So it's nice because it kind of keeps the idea of a summary statistic, but you also can see the kind of full distribution of values just to make sure you're not missing something by taking summary. Similarly, again, with the histogram, we can plot a kernel density estimate of the kind of distribution. So it just goes a little bit further than the tools that you have with Matplotlib, for example. Then everyone's favorite, the heat map. So this is really nice for displaying matrix style data where you have kind of paired data along an X and Y axis. It's nice for functional imaging applications. You can show areas that are polarized, positive and negative, for example. So you can choose to have values on it, but obviously you have this kind of bar alongside that is a color indicator. Then you've just got the standard scatter plot, but obviously, again, it has a few more dimensions to it. Then you have this swarm plot. I haven't seen this before now, but I had a look at what it does, and it's essentially very similar to the violin plot, but you can actually represent two dimensions here. So you have the distribution is kind of represented by the kind of silhouette here, but every data point also has its own representation on there. So you kind of avoid over simplifying the data as well. Okay, so now we're kind of going to come on to some of what you can do to extend these tools beyond the kind of basic libraries. One really important thing that I think gets overlooked maybe at undergraduate level is not to just use kind of your basic red, black, green, orange for plotting. So Matplotlib, for example, has this full library of every color you can basically imagine. And I would really recommend having a look at these once or twice just to kind of get an idea as to color schemes that can contrast your data a bit better than the very sort of neonized reds and blues that you get in a basic packages. So aside from that, there are certain situations where we might want to customize beyond the kind of discrete colors available in Matplotlib, etc. So I'm going to show you an example of how you can customize your poster or plot or whatever it is that you're trying to change, just using the basic Microsoft tools. So I'm going to attempt to show this outside PowerPoint. Okay, so for instance, if I want to create a shape and match it to a specific color, I'm going to start by making a shape just in my PowerPoint. Okay, let's choose a heart shape, for example. Okay, so now I can choose the color of my shape. Okay, I have the basic preset ones, obviously. If I go to more fill colors, you have this kind of pipette style color picking tool. So if I click on the tool, I can basically select a color from anywhere around this. So one nice way to do this would be to bring up this diagram and I can select something that matches my plot. But let's say I want to match to the King's logo. I will just click and select OK. And then I've matched the exact same shade. So it can be nice for kind of consistency and cohesion in your plots. Another thing that you can do if you want to code it, for example, so this is what I've tried to kind of represent over here, is you can actually code colors that you prefer using the RGB or the hex codes. So this is what I've been doing to plot kind of brain atlases, for example. But you can do it in Microsoft just by going to this RGB sliders tool. So it's kind of on here. And it will actually tell you the RGB code, for example, of the King's logo. So this is something that companies really tend to use when they're making presentations for clients, etc. Most companies will have a kind of RGB or hex code of their logo that they tend to use like across all of their presentations, for example. Okay, so you can also take the hex color from down here. Brilliant. Let me come back onto here. Okay. So I've put together some examples of color palettes that you could create using the Matplotlib basic colors. This one I tend to use for mine because I'm looking at sort of 4 behavioral measures, for example, and just throughout I will use one different color for each one. It just helps to create some consistency. Okay. Great. So this kind of brings me on to this really cool statistician, Edward Tufte. So he's a statistics and computer science professor at Yale, but he's also produced a series of, I think, 4 books which cover data visualization and statistical representation. And his basic area of interest is looking at sort of through history, different ways that data representation has been used to kind of change the course of scientific research or change the course of history, etc. But I just wanted to kind of take his 4 key principles related to data visualization. I think that some of them are kind of a legacy of times gone by when we think about kind of ink maximization and things like that. But the first one would be this idea of graphical excellence. So his idea is that the primary purpose of representing data is that we want to communicate sometimes complex ideas, but with clarity, precision, and efficiency. So by clarity, we would mean like it's really evident what we're trying to show. Then precision, it's talking about we're showing something that's accurate to the data. We want to represent data in a way which appears obvious. And by efficiency, we just mean not wasting too much space or time thinking about aspects that don't really represent the data very well. The second principle is this idea of graphical integrity. And I was kind of interested by this when I was reading about it, because I think, you know, in the period of sort of more AI and data generation and things like that, I think our generation can be, maybe rightly so, quite cynical about whether the data that we're being shown is really accurate or whether it's misleading or we're kind of having the wool pulled over our eyes in a sense. So I think it's really important to remember the kind of purpose of data presentation. It's not to kind of deceive an audience as to a conclusion that you hope is the case. It's really to represent a real relationship within data that you work with. So we're not just kind of coming up with a hypothesis and trying to stress it with an elongated box plot or trying to kind of stress a relationship with a scrunched up scatter plot, for example. We just want to represent data that really is there. So I think it's something to think about as you go through your work. The third point is a kind of legacy of hand drawing, but there's this idea of data ink maximization. So this kind of reflects the idea that on the page of your plot, the majority of the ink, if you like, is contributing to useful data that is representing your point, as opposed to kind of unnecessary elements outside it, for instance, grid lines, etc. And that kind of links to the final point, which is this term that he coined, chart junk. So a lot of us will start a plot by just kind of coding a title, axis titles, X and Y axis ticks, a legend, the grid lines on, etc., etc. But sometimes this kind of can distract from what we're trying to show rather than add to it. So I've got an example of a plot which has a bit of chart junk surrounding it. Okay, so feel free to shout out, but what features of this chart do you think Tufte would consider to be chart junk as opposed to useful data?
The extreme greed.
Yeah. Okay, yeah. The grid is pretty busy. Anything else? Yeah, exactly. It's pink for one, not probably necessary. And also, you don't always need to have a border around your plot particularly. Any other things that we notice? Yeah, that's not really helpful here. And a lot of the time you have a side plot which is explaining the kind of color scheme anyway. So it's not always necessary to list everything out in a legend.
That's too many.
Yeah, we probably don't need, in fact, any information. I mean, what this plot is trying to convey is a kind of difference between distributions in a brain regions, for example. If the numerical values are not really that interpretable, it's not necessary to have them unless it's relevant to the point that you're trying to make. So here, if we're just trying to represent difference, it's fine to leave those out or just have kind of anchoring points that remind us of the numerical value. Apart from that, I think unnecessary, just color generation, it can be distracting rather than useful. And when I was reading the book, I kind of, he has a section where he's talking about the fact that data visualization isn't a kind of art project. It's really supposed to elevate data for audiences rather than be a kind of outlet for your personal creativity. So this is an example of cleaning it up slightly. We can see, okay, I can now see the kind of differences between the distributions for myself. And I think it's a nice kind of way to think about data representation, is that you want to make what you're showing obvious to audiences. Yes, they'll believe you if you show them a p-value that's significant, but I think it's a lot more comfortable if they can also see for themselves that there's a difference in data, for example. And people are a lot more likely to trust the kind of graphical integrity if you just really make it obvious to them. Okay. Someone who does this kind of principle really well is Isaac Sabinius, who's another ex-PhD student working with my supervisor. And this is just like one figure from his paper about using morphometric inverse divergence. But what I really like about what he's done here is that wherever possible, he has removed kind of uncontextual data from the plots and really just stripped them back. I can't really think of any other way you could have 8 plots in one figure that doesn't kind of overcomplicate and confuse. So for example, 2 things are shown on one plot, but it's just a simple color scheme throughout with a basic key of purple and green. And I think even the color choice of a quite pastel color is kind of nice. It doesn't really distract from the distributions, for example. Another choice that he made is here we don't have kind of excessive scatter plot drawn on. It's just the best fit line that is kind of the point that's trying to be made. So I think we can all kind of take a sort of message of stripping back some of the plots to not have excessive detail. Sometimes things like grid lines are necessary, but there's kind of creative ways that you can incorporate them without sort of creating this grid over your plot. Okay, so the next thing that I'm going to kind of move on to beyond the basics that we've talked about now is when we want to plot anatomically relevant data. So I think for a lot of people here, it's going to be useful to think about data that's inherently kind of tied to the anatomy that we study. So for us, it would be the brain, sometimes if we're working on the heart or vessels, etc. So a lot of the statistical data that we work with is kind of inherently tied to the basic anatomy. For example, maybe we're looking at kind of physical features of the brain or something like that. We might have arterial wall thickness if we're looking at vessels, or we might have kind of sulcal depth or cortical thickness on the brain. Other things like comparative regional measures, it's fine to have a box plot comparing regions or a violin plot like we just saw, but a really nice way to stress this kind of thing is to plot on the actual anatomy. So maybe you need to change the color of the left ventricle compared to the right ventricle. Other data that we work with is slightly more complex things like functional metrics or kind of connectivity measures. So things like fMRI produces like activation maps. It's useful to look on the actual brain. And we can sometimes see these kind of interactive videos of polarization across the brain as well. Okay, so the kind of data that we work with in terms of anatomy is usually either confined to one of surface or volumetric. So with surface data, we're dealing with things like a mesh with vertices and faces and so on, whereas volumetric data is more kind of linked to voxels, for example. Okay, so I want to kind of focus in on slightly more complex data that we work with, which is this concept of a connectome. So you can take it more or less abstract depending on the application, but a connectome essentially is a kind of graph that represents the connectivity between anatomical regions in this case. So what we mean by connectivity is some kind of relationship between the two. It may be functional connectivity, regions that fire together, for example, in the heart, regions that are experiencing an impulse at the same time. Or it might be a connectivity measure like similarity, for example. Okay, so I've done an example of a kind of anatomical graph, or a connectome, if you like. But what organ is this? Any ideas? Why is it not obvious? Because this is just a kind of uncontextual graph, right? It doesn't have a kind of anatomical relevance, and we can't tell what region this is, for example. So when we plot connectomes, it's useful to also be able to plot them on the relevant anatomy rather than just in kind of free space or in matrix form. Okay, so some of the libraries that we have for things like plotting on a anatomy would be I'm going to focus on neuroscience and plotting on the brain at the moment, but some of these libraries are also applicable for the heart, etc. So for instance, we have packages like NiLearn, where we can plot kind of regions of interest, like parcellation maps, so they show different regions of the brain. We can also plot statistical maps. So this would be things like regions of activation from like fMRI, for example. And another similar one is this kind of glass brain idea where we can sort of plot a three-dimensional perspective through the brain. Yeah.
And the brain, or do you have to provide them?
So you can use kind of standard packages that they have, but you can also input your own surfaces or brain volumes, for example.
They require
Yeah, they work with like nifty and gifty formats, so .nii or .gii files. But I think that you can convert to those if you have data in other forms. Okay. Another thing you can do is outline regions of interest. Maybe this is more useful if you want to still keep some kind of contextual information about the surface inside. Then we have PyVista. This kind of has mixed reviews, this one, but it's essentially a helper library for the visualization toolkit, the VTK. So what you can do with PyVista is you can construct 3D plots, but they kind of have this interactive window, so you can scroll around the brain, for example, around the heart. I think it's difficult for situations like displaying on a poster, but if you really want to like interpret your own data, it's nice to be able to scroll around it and see what's happening rather than just always being confined to the kind of three basic views in the context of the brain. Okay. So a kind of interim step, if we want to look at a connectome, rather than just always plotting on the brain, for example, a kind of cohort comparison that we can do. Instead of plotting everyone's brain individually, we can just plot on a kind of connectivity circle. So we preserve the information that relates kind of which regions are connected, but without the kind of spatially complex nature of plotting inside the brain. So what they do is you can kind of input your matrix, for example, your connectivity matrix, and then you can specify what the kind of nodes are going to be. So in the context of the brain, this would be your parcellation regions, for example. Then what we can do is we can just plot on a circle different regions that are connected to each other, and you can have the kind of strength of your matrix value as the color map, for example. So this was using a library called MNE connectivity, but I think other ones are also available. This is just an example of how I kind of reduce the dimensionality of it just to look at which regions are more connected, for example. So you can play with those and see different relationships within your data. In this case, this is just a representation of the different regions that are connected. You can take this to its extreme. This is one example I've found where they have done that. Okay, so this is also a connectivity circle plot. This actually shows 8 different dimensions of data. So this is representing the brain as well. But not only do we have the kind of standard connectivity matrix going on inside, it has 3 different colors to represent different polarization. Then we also have parcellation regions, general and specific, around the outside. And then these metrics are for surface area, cortical thickness, and so on, and they're represented around the outside. I think this is more useful as a kind of lookup tool rather than a kind of obvious description of what's going on the brain surface. But it's nice to see in one place a kind of representation of a brain that we might want to use. Okay, so another thing that I want to mention is that, yes, we have these plotting libraries like Connectivity Circle, like Matplotlib, and like Seaborn, and so on. But you're not restricted to just using those libraries. So if you're working with brain data, for example, you can play with different plotting tools that are available and kind of come to your own conclusions. So one example of a brain plotting tool is Connectome Workbench. So this is like the tool that we use to view brain volumes and surfaces. You can plot regional parcellations on here and so on. So what I have done when I want to plot regional values on the brain, for example, regional strength of similarity, is every parcellation region has this kind of .anot file that indicates which vertices are related to which brain region. And what you can do is you can just reprogram these label files so that instead of corresponding to the parcellation region, this is where you plot your kind of regional value. So you plot your heat map value instead of a parcellation region. And what ends up happening when you plot those on the brain is you can see the kind of distribution of regional values from your heat map rather than a parcellation region. So I think this is a nice way to look at, for example, regions of importance from a prediction model. So this is what these are. So when I run my prediction model or whatever it is that I've been working on, I might want to have some kind of interpretability about which regions are most relevant for predicting X, Y, or Z. And what I can do is while I run through, I kind of keep a track of which vertices and which regions are important. And then I can plot the value on the brain like this using whichever heat map I choose. This one is magma. And what it just means is that we can see it in the context of the brain anatomy. So it's not on a circle and it's not just kind of list or a matrix. Okay. Then if I want to plot something beyond a kind of regional summary metric, we can use something like PyVista to plot our brain surface or the heart surface or whatever anatomy you're working with. And I've just come up with a quick script to plot the kind of location of regional centroids. So my brain is parcellated up into roughly 68 regions. And in the center of each region, which is just quite simple to calculate, I just plot the kind of node, so the central point. And then I can plot my connectome using kind of basic pipes that join up each of the nodes. And what it means you can do is you can kind of see in the context of the anatomy what the sort of connectivity graph looks like. The nice thing about PyVista is that it has this 3D visualization window. So you don't have to take my word for it because I can show a quick example. So this is just a script that I was showing on that previous slide. So once I have my connectivity matrix, I can basically run a quick script to just visualize the connectome. And let me just bring it onto the bit you can see. Okay, here. So what I get is this kind of 3D interactive window, which shows my connectome fully on the brain surface. So here you can see the kind of two hemispheres and the connectivity matrix. I think we can agree this is hard to interpret. So what you can also do is you can look at slightly lower dimensional version of your data. For example, here I can just select a threshold and it's only going to show me connectome edges which are kind of the strongest. So if I run it again, I can quite quickly see. Yeah. Yeah, so you can change the thickness of the pipes, you can change the color map, for example. So when I plot the strongest edges of my connectome, I can immediately see the majority of what's kept are the bilateral edges between the two hemispheres. So just with a quick plot, I've already got kind of contextual information about the location of key edges beyond plotting them on a kind of 2D axis and looking at where they emerge. So this is just something that I've kind of worked by coding my own node and edge pipes, for example. And I've added in something here which can basically locate the key hubs, so where the predominant connections are. And I've just represented them with larger radii, for example. Then I just had a play around. I've changed the color of the nodes, for example, to match the kind of average regional connectivity. So darker nodes represent stronger overall kind of regional connectivity. So there's heaps of stuff you can do just like using PyVista or other plotting libraries, but adding a bit of innovation by just adding your own kind of connectivity matrix, for example. So I'll share the slides after this if anyone wants to kind of replicate that. Okay. Brilliant. But I think this is a really nice way of kind of using data visualization to draw your own conclusions in your data. So not just to kind of convey it to audiences. Something like this probably isn't going to be as meaningful in like a conference type scenario, for example, but for your own kind of research into the statistical distributions within your data, I think it can be really helpful. So somewhere where I've used this in my work, for instance, is in combination with another plot. Okay, so something that I wanted to look at was the kind of age trajectories of different points in my connectome, so different edges, as in lines in my connectivity matrix. So what I've done is I've started with the kind of 2D plot that you might use. So this is just tracking kind of edge strengths across age. So you can already see a few things that we've already talked about. For instance, this kind of error window around each line. So this is nice for plotting trajectories because you can kind of see the general trend. For instance, here, you can see from the green kind of window that some are starting much lower and increasing more. Then what I've done to kind of contextualize where that is on the brain is I've isolated which edges this was talking about in my connectome. And then I've plotted those edges on the brain using PyVista. So the kind of key hubs are shown using the nodes, and then I've got the kind of full connectivity of edges in between. So just from doing this, I could start to see that I have an increasing trend in similarity around the kind of lateral regions of the cortex, whereas more consistent features are found kind of within the medial sections and vice versa for the occipital regions. So I think this is a nice example of kind of combining the two concepts of a kind of traditional 2D plot that you can look at and also contextual sort of anatomical information. Okay, so now I've kind of covered how to plot connectomes, for example. I'm going to move on to talk about the kind of context in which we want to convey research to audiences. So things like grant proposals, poster presentations at conferences, even kind of on slides within a kind of presentation talk. So I'm going to start with talking for a minute about poster design. So this is an example of a poster that I made for OHBM last year. So it's a brain mapping conference. I think it's important to think about the kind of purpose of presenting a poster, for example, at a conference. For those of you that haven't been to a conference before, the nature of a poster presentation session is that there may be kind of up to 1,000, even at big conferences, posters just sort of laid out in corridors around the room. And you stand by your poster and people will come up and read them and ask questions and so on and so forth. So you need to appeal to a real kind of variety of customers, if you like. So one type of person is going to come up to your poster and just read it for a bit of light relief because they've been at the conference all day. They just want to have a look at what you did. Maybe they don't even work on brain mapping at all and they're doing something else, but they're just a bit interested. So this kind of person, you need your poster to be kind of accessible to them to just have a quick skim through, and they're not necessarily going to understand the full methodology of what you've done. Another type of person is going to be someone that's working in an extremely close field to this, and they want to understand the full pipeline, every analysis that you've done, what your results are, kind of why it's interesting. Yes, they can speak to you if you're at your poster, but you kind of want them to be able to draw their own conclusions from the data first so that you can have a kind of good conversation about what you found. So the level of detail can be difficult to kind of decide whether you want to include every piece of information about what you've done or whether you want to kind of keep it really visual and simple. So I would say that the key aims of a poster is really to grab passing attention. You want people to skim across the row of brain posters and be like, this one feels accessible for me to read quickly and understand. Maybe you're jet lagged, maybe you're a bit tired. it's easiest to read the posters, which are more kind of visually obvious. You want to display your results. Again, this kind of touches on what I mentioned before, that people tend to be a little bit egotistical, and if they can already get a sense of what you found, they're more likely to interact with you about your results because they might feel they have something to add. And these are actually the useful conversations to be having at a conference. Beyond that, you just want to kind of highlight the key information. So you don't want everyone who comes to your poster to be asking you about how many participants there were. It should be somewhere on your poster. You want to kind of keep the missing information just for really interesting kind of scientific conversation rather than discussing the methodology, for example. So my top tips for making a conference poster would be to start with a template. I had a go at making a poster from scratch the first time and it looked absolutely terrible and I'll never try that again. But you can find really simple templates somewhere, like this website. It's called posternerd.com. If that doesn't put you off, I recommend it. And King's have templates as well, so you can get a kind of standard conference poster type template from King's as well.
Take it from King's.
Yeah, exactly. Obviously, you don't want to kind of tread on the toes of your colleague, but a basic structure is really nice to have. Otherwise, it can feel quite an overwhelming task to kind of present your data like this. Okay, so beyond that, I think choosing a content appropriate color scheme can be nice. Like a lot of people tend to keep to a somewhat monochrome style in a conference, sort of black and white, maybe a bit of red if you're lucky. I've been thinking about the kind of relationship between color and sort of conceptual meaning. And I think that it's quite a strong thing that we have a sense of. For instance, like I remember in school when we would be given like a exercise book to write in for maths or English or whatever it was. People I knew from different schools would always have such a strong idea that like maths is a red book subject and like English is a yellow book subject. And I think that our relationship with color and kind of concept is really stronger than we maybe give it credit for. So this was a poster about kind of neonatal brain development. I decided to use a kind of pastel color scheme. It has links to this kind of idea that we're really working with babies, for example. And in a conference full of work across the lifespan, there's lots of posters to do with Alzheimer's and Parkinson's and things like that. I think this just kind of reminds you that this is the kind of context of what we're talking about. It's really early life development. And it can be nice to bring out kind of emotive links as well to the importance of studying the neonatal brain. So I would avoid large chunks of text. In my experience, you should kind of imagine that your average conference customer is quite lazy. They maybe don't want to read a whole essay every time they come to a poster. And it's going to be quite small. So you want your text to be large and quite succinct with the key detail, obviously. Okay, so the key considerations that you might make beyond that is who the target audience is. So not every conference is just going to be academics, for example. What are some other kind of conference audiences that you might have? Yeah. So this is a really good example of a type of audience that we need to kind of tailor a poster to. If we go to a clinical conference, the kind of things that we want to be stressing are things like useful pipelines that they could incorporate in a clinic or results that are going to impact the way that they run their clinical analysis, for example. Clinicians are really interesting people to talk to about the kind of impacts of our research, but you're going to lose them if you have heaps of mathematical pipeline that they're not interested in, frankly. So if you go to a clinical conference, your poster is going to look different to if you go to a kind of really research-focused conference. Likewise, if you go to a research conference with a kind of really simple, basic result that would suit clinicians more, you're going to lose your research audience because they don't feel like they can follow and replicate your pipeline, for example. What other kind of audiences could we have?
Students.
Yeah, okay. So this is kind of similar to a research style audience, but the level of detail and the level of mathematical content needs to be a lot less and a lot simpler. People are going to engage more with something that's obvious to them than something that kind of goes a lot further beyond their understanding. So you want to kind of grab their attention with something that they can engage with and then build up their understanding kind of throughout the poster.
Probably mostly you're pitching to an audience of your peers, right? Because like the academics often are off in other meetings and stuff like that. So you're trying to pitch to some people that won't have lots and lots of background knowledge necessarily.
Yeah, exactly. Yeah. And I think you can make up some ground with having a really good pitch if you have like a really simple explanation for what you've done. But it saves you a lot of effort if you already have like really simple figures to refer to. So it kind of depends on the context of your conference. At OHBM, I knew that I was going to be sending at the poster for the sessions, and I knew that it was quite a packed schedule within that. So I've made it so that I can kind of use it as just a visual reference to speak to people using, as opposed to something that's going to be left there and everyone to read it kind of in their own time. Another really important consideration to make is this kind of idea of making research more accessible. So it's really quick to make your poster just basically accessible to the type of people that might be reading it. For example, don't use minuscule font sizes, especially if you're pitching to kind of like patient cohorts or if you're kind of representing your data in a sort of setting with people who might be nearsighted or older people. It's really important that your poster is basically readable. Another really simple thing to check, which maybe you would look past, is to just run your work through a basic colorblind filter. So you can find these online just for free, and you just paste an image in there. Maybe you have something like this, where you've got different brain regions in color. If I run this through this color blindness filter, I can quickly see what people with different types of color blindness are going to see when they look at my poster, for example. If these two look the same, then I would recommend potentially having a colorblind test. So this kind of shows that the color scheme I use for the brain, at a basic level, we can really distinguish between the different regions irrespective of the color blindness test. So this is going to be a really nice kind of color scheme to use, for example. If you've got really subtle differences in hue between like red and brown, it's going to be a lot less accessible for people. Okay, so finally, if we think about the kind of design elements of a poster, just by stripping back some of the detail, it makes it a lot harder to kind of orient myself when I come to read this poster. So what you want to kind of draw the audience's attention to is things like the introduction and the conclusion, and then a kind of path through your figures. So just by adding some kind of background gradient, just by including kind of key boxes and so on, I can sort of draw my audience to the parts that I want them to notice first. So these are some kind of key things to note when you make a poster. Again, I've talked about the content appropriate color scheme, but most posters have the convention of a kind of 2-column layout, notwithstanding exceptions. Avoid putting black or white kind of stark contrast figure backgrounds if you don't need them. I appreciate a lot of anatomical kind of visualization tools like Connector and Workbench. They come with a kind of black medical imaging background behind every brain plot, for example. But it just takes a simple background removal tool in PowerPoint, for example, and then you can get these plots that are kind of in isolation. So I think this is a nice way to avoid there being this kind of excess white everywhere on your poster. Numbering section headings is useful. It might be obvious to you because you made it, but it's just a quick way to kind of take people around the poster. And then don't forget to include things like funder information, but also you can have a QR code that has a kind of PDF version of your poster. It's nice, some people will scan the QR code and just zoom in a bit if they're not able to come closer to your poster. So I think that's really nice. And you can hand out a basic kind of business card with the QR code if people are going to find it useful. Okay, so when you're working around the kind of poster, these are the sort of key elements that you're going to notice in sequence. So the first thing they're probably going to notice is the title. So that needs to be kind of stark contrast. Try not to use kind of yellow on purple or things that are not going to print well. But some kind of contrast, large font, you want people to read that from quite a long way away. Then the first thing I would usually come to is the introduction. This is just going to anchor the audience as to what your poster is really about, basically. So that's the first thing that I would read. Then if they're in a rush, the next thing they're going to read is the conclusion, basically. So this should be like a really quick summary of what you actually found. And I think it's nice to kind of match the two across. Then someone who has a bit more time is going to read through the actual content of your poster. So they're probably going to start with number 1 if they're doing what you've asked. Cool. So the first thing I'm going to then come to is the first figure. And this should be like in chronology with the work that you've done. So if the first thing you've done is kind of plot one example network, it makes sense. Then subsequent analysis is going to come later. And so on and so forth. The next thing they're going to see is going to be the next plot, and then they're going to come up to the right. You can do your posters kind of left to right, but it's not the convention, so it's more likely that people are going to be reading down and down. Okay, so finally, I'm going to talk about pipeline flowcharts as the last thing. So this is kind of something which I use to design aspects of my PhD work. You can use them in posters, you can use them in grant proposals and things like that. It's just a way to represent your kind of methodology in a visual way rather than a large paragraph chunk of text. Okay, so I'm going to show you this pipeline flowchart that I've made. This is just a kind of proposal for an aspect of my research. And this has gone in my upgrade report, for example, so I can kind of launch the research that I'm going to do next. So I kind of want to try something slightly experimental. Can I have someone volunteer to come and see if they can describe what the pipeline is kind of involving just from the flowchart? Any volunteers? Or I'll pick Nishira. It's your last chance. Super. Okay, do you want to give it a go?
Okay.
Okay, so Nishira is going to try to explain roughly what my pipeline involves just from the flowchart.
So this pipeline takes in two inputs, MRI images and some more features derived from the images directly on surfaces. And it's got some learnable feature weights that the pipeline is going to work on. You're going to have a run through of your input data through your weights, and that's going to give you some predicted regional feature metrics and interesting maps, which is going to go then into a section that says graph neural networks for predicting behavioral outcome predictions. So this is going to take this part as inputs, and it's going to put them through a few networks, and the output is going to combine with some behavioral outcomes, which I imagine that's the ground truth data. And here's your loss function. That's going to compare your predictions to your ground truth, and that's going to update your weights and go again.
Great. So just from absolutely no contextual information and no relevant information about my methods, Nishira basically can describe what my pipeline is going to involve just from the flowchart. Yeah, you're not going to have this in isolation normally. You're going to have a description of what's going on. But I just think it's really nice to be able to kind of show it all in one place. Maybe there's small details that they're not going to get from just the flowchart, but I think it's a really nice way to kind of support a kind of grant proposal or a research plan, for example. Another reason to make flowcharts that kind of explain your or depict your pipeline is when I'm making these, I will realize that one aspect of the flow doesn't make sense, for example, and then I'll move the blocks so that it makes more sense to run it that way. So I think it's nice to see kind of throughout different data visualization techniques. Not only can we use it to convey to audiences, but to kind of improve our own understanding of the work that we do and our own analysis of the work that we do. The kind of things that you can put in your pipeline is like basic kind of input information. You can label blocks that do different things, like maybe you have your surface pipeline and you might go into more detail somewhere else in your proposal, for example. Then you can kind of combine it with these sort of aspects that you see in the state space type models where you take predictions and ground truths, for example, it's nice to see the loss function, and then you can kind of plan your pipelines like this. So I think it's something that's worth doing. There's other tools you can use to do this for you, but it's also quite simple to just make it in PowerPoint. And I would really recommend having that to kind of add to a proposal, for example. In the context of deep learning, you're probably going to have an actual specific deep learning architecture that you keep in your poster, but some of the same principles apply. Okay, so I'm just going to summarize what we've kind of seen so far, which is that data visualization is important and it's meaningful. It's not just a kind of pretty picture to go alongside your research. It's a way to prove to your audience that you have found something genuine and real. Another thing I think is important that really was impressed upon me by reading these books by Edward Tufte is that published figures now that maybe you publish in your journal paper or maybe you're publishing in a conference paper, these become the historical archives of our current scientific knowledge. So now it might feel like everyone's publishing a brain plot and everyone has this kind of figure or that kind of figure. But in like 100, 200 years' time, these figures are going to be the kind of information that contextualizes like the current scientific knowledge that we had now. So I think it's really important to see what we do as really like important foundational work. The same way we have this timeline that shows the kind of first coxcomb diagram that was used to represent war fatalities, for example. Someone could just as likely be using your data to show the kind of understanding of the brain that we had in 2020, for example. Another key sort of takeaway is that less can be more. So yes, it's important to add contextual information and show multidimensional data, but let's not have dense grid lines and unnecessary legends and so on. Then in terms of anatomical data, I think we can kind of see by now that plotting on anatomy gives a lot more contextual information to a kind of statistical plot. And finally, I would stress that data visualization isn't confined to the conventional libraries. So even if you work with a tool like ChatGPT or something like this, you can get ideas for how to plot things. If you know what you want to show and you feel like the current libraries aren't doing it, just have a bit of a play with tools that are available and see if you can come up with something. This is how new data representation methods get made. Finally, I think intention first, data representation is something important to keep in mind. So it's not just plotting your statistical data for the sake of it, but you need to have an understanding of what aspect of your data you want to stress. So do I want to stress a comparison, in which case I need to plot something like a box plot to just compare summary metrics? Do I want to stress a kind of distribution of data being really broad, for example, in which case I'd use a histogram? Lastly, you may not be your own target audience. So just because the figure makes sense to you, it doesn't mean that this is going to be most appropriate for a kind of conference audience, for example. So just keep in mind who you're kind of showing your data to, whether it's your supervisor in a weekly meeting, whether it's a kind of patient type conference audience. Effective data presentation makes what you've done look obvious. We were kind of talking about this earlier, that sometimes in papers, for example, people will try to make the work they've done look extra complicated so that it seems more impressive. But the research that people engage with most is the research that looks obvious. You want them to think, why didn't I think of that analysis, for example? Or like, I can use this to improve research. The idea of what we're doing is not to kind of gatekeep our methods. It's more to build scientific knowledge, and we want to kind of keep that accessible to other people. So I'm going to leave you with this kind of inspiration slide of some of the plots that I've made for my PhD upgrade, so you can see some kind of examples of how colorful and visual data representation can really sort of elevate the research that you do. Thanks for listening. I'd love to take any questions if you have them.